Hello students, welcome to the EPG Pathshala. My name is Dr. Rajeshwari Sinha and today in this lecture we are going to learn about the primary structure of proteins. In today's lecture we will study that primary structure means the amino acid sequences in the polypeptide chain which is linked with peptide bonds. We will further do an in-depth study of their function and underlying biochemistry. Later on, we will also explore the significance and application of peptides and proteins. Let us see what are the various objectives that will be covered in this lecture. First, we will understand an introduction to primary structure of proteins, the basic units of proteins as amino acids. We will look at the underlying biochemistry of peptide bond formation process. To know about the features of protein backbone, we will also look at the relationship between genetic codes and sequences of proteins and other significant amino acid sequences. We hope you have an enjoyable time listening to this lecture. Introduction. Proteins are very important functional molecules and participate in almost every biological phenomenon. They are playing important roles in the form of enzymes, structural framework, muscle movement, membrane function in control flow, sensors, switches, gene function, regulation, signaling, DNA replication, transcription and translation, etc. It has 3 or 4 hierarchical structures such as primary, secondary, tertiary and quaternary structure. Functionality of protein arises from its tertiary or 3D structure. The simplest one is primary structure, which is basically a sequence of 20 amino acids and all other hierarchical structures that are based on specific amino acid sequence of the primary structure. All information regarding the configuration, function and position of protein is written in the language of sequence of these 20 alphabets amino acids. Amino acids as building blocks of protein structure. Amino acid composes proteins and are of 20 types of monomeric units. Each amino acid is comprised of 4 different chemical groups bonded with 4 valences of alpha carbon atom, 1 amino NH2 group, 1 carboxyl COOH group, 1 hydrogen atom and 1 specific group called the side chain. All amino acids are having the same general structure, but they are different from each other on the basis of specific side chains. On the basis of solubility, nature of side group of amino acids, they can be categorized into 3 groups: hydrophilic, hydrophobic and special amino acids. Peptide bond formation. Proteins are composed of amino acids ranging from 50 to 2000 linked in a chain with peptide bonds. Peptide bond forms when 2 amino acids make covalent bond in between amino group of 1 amino acid and a carboxylic acid group of adjacent amino acid on taking out 1 water molecule. We can see the reaction below. This reaction remains in equilibrium in the direction of hydrolysis, therefore requires free energy. Kinetically, peptide bonds are quite stable and lifetime of this bond is about 1000 years in aqueous solution in the absence of any catalyst. Amino acid sequence. The sequence of protein is written by using 3-letter abbreviations of amino acid residues. This primary structure is usually shown using abbreviations for the amino acid residues. For example, pro, arg, val, tide, gly, lys, pro, glu, tri. Polypeptide chain has 2 ends at both sides of different polarity. One end is with an alpha amino group and a carboxyl group at the other. An amino end is the beginning on the basis of translation. Therefore, sequence of it is written in the direction of amino to carboxylic end. Hence, amino acid sequences are directional. This continues from what we have been talking in the previous section, that amino acid sequence is directional. It moves from the amino terminal residue to the carboxyl terminal residue. This figure shows the directional nature of amino acids in a polypeptide chain. Chemical characteristics of polypeptide chain. Proteins are composed of about 50 to 2000 amino acid residues, and peptide chains having less than this is known as oligopeptides or simply peptides. An average weight of any proteins may vary from 5500 to 220,000 Daltons. On the basis of this weight of one amino acid comes about 210. In the polypeptide backbone, C double bond O and NH groups of peptide bond are repeating regularly throughout the protein and play an important role in the formation of regular structure or secondary structure throughout the hydrogen bonding. Chemical characteristics of polypeptide chain. In this, all electronegative oxygen of carboxyl group and hydrogen of NH are engaged in hydrogen bond formation with each other and with functional groups of side chain. Therefore, a regular structure emerges out and provides structural stability to the protein. Whereas side chains of these amino acids are protruding outside, and depending on their nature, different interactions are taking place with the surrounding atoms. Disulfide bonds. Apart from these regular hydrogen bonds, other types of non-covalent bonds form, like if cysteine residues are present, then disulfide bonds form in between two cysteine residues, resulting in linked cysteine, also known as cysteine. The figure alongside shows this reaction. Terminology in peptides. On the basis of the number of amino acids present in the peptide chain, it can be termed as dipeptide for 2 residues, tripeptide for 3 residues, oligopeptide for 12 to 20 residues, and polypeptide many residues, and proteins for 50 to n residues. Proteins having more than one peptide are known as multimeric proteins, and when they are identical, it is called a homomultimer, and if the polypeptide chains are different, it is called a heteromultimer, for example in case of hemoglobin. So students, we hope you are finding this lecture useful. We are looking at the primary structure of proteins in this lecture. We have seen that amino acids are building blocks of the primary structure of proteins. We understood how a peptide bond is formed and also delved into understanding some properties of amino acid sequences, disulfide bonds, and chemical characteristics of peptide chains. In the following part of this lecture, we are going to make an effort into understanding this peptide bond in more detail. We are going to see its spatial positioning, the different bond lengths, alignment of the different bonds, and functioning of the protein backbone. We are also going to introduce the concept of phi and psi angles and Ramachandran plots. Features of the protein backbone. The protein bond or the CONH bond is a rigid and planar structure in nature. There is no rotation occurring in this bond, and all molecules of the peptide bond, which is alpha carbon, carbon, oxygen, nitrogen, hydrogen, and alpha carbon, are present in the same plane, and the side chains lie above and below of this plane. The figure below shows this planar structure of the peptide bond. On analysis of the bond length between CO, CN and NH, it is revealed that the bond length between CN in peptide bond is 1.32 angstrom, which is in between a single 1.49 angstrom and a double bond 1.27 angstrom of a CN. It is concluded from this study that peptide bond exists in 2 resonance forms shown in figure. Due to this, the peptide bond has a partial double bond character, which makes this bond rigid and nonpolar. This property facilitates folding and attaining globular structure to polypeptide chain. This slide shows the different bond lengths of the peptide bond. We see that the bond length between alpha carbon and carbon is 1.51 angstrom, the carbon and oxygen is 1.24 angstrom, carbon and nitrogen is 1.32 angstrom, nitrogen and hydrogen is 1 angstrom, and nitrogen and alpha carbon is 1.45 angstrom. Resonance structure of peptide. The figure shows the peptide bond resonance structures. Resonance increases polarity of peptide bond and therefore the dipole moment, due to which the hydrogen bond forming capacity of the backbone increases, and polarity also plays an important role in protein function and its behavior. Configuration of the peptide bond. The planar structure of the peptide bond may attain cis and trans configurations. Almost all proteins have trans configurations in peptide bond. In this trans configuration, the two alpha carbon atoms are on opposite sides of the peptide bond, and while in the cis configuration, they are on the same side of the peptide bond. In trans form, steric hindrances in between groups attached to alpha carbon atoms are lesser than cis form, as they are not present in the same plane on the bond. Like we have been talking before, this slide gives a pictorial understanding of the cis and the trans configurations of the amino acids in the peptide bond. The rotation of the phi and the psi angles. The angles of rotation between the single bonds of the nitrogen and the alpha carbon atom and the alpha carbon and the carbonyl are known as phi and psi respectively. When these bonds rotate in clockwise direction, then their values are considered to be positive. Ramachandran plot. Professor G. N. Ramachandran proposed about the feasibility of different possible combinations of these two angles, phi and psi, with respect to stability of corresponding protein structures. All combinations of phi and psi are not possible to make a stable protein. Therefore, a particular combination of phi and psi angles are required in backbone of peptide in order to get a stable and specific structure of the protein. This slide talks about what we have been discussing before. It shows how different angles of the psi and phi can be combined to get a stable and specific structure of a protein and also a disfavored structure of a protein. We are going to discuss Ramachandran plots in detail in the upcoming modules. For now, we are going to dedicate the next section of this module to understanding the importance and functions of amino acid sequences and their positions in primary structure. It will be interesting to see how and where simple amino acid sequences are used, what information can be derived from them, and in what areas they find application. Let us take a look. Amino acid sequences coded by genes. Each protein has its own specific amino acid sequence, which is defined by the genetic code of DNA. This was deduced by Frederick Sanger, who is the first scientist who determined the amino acid sequence of hormone insulin in 1953, which is a landmark in science, and now amino acid sequence of more than 100,000 proteins have been determined. It was revealed that amino acid sequences are defined by genetic codes of DNA and which carries hereditary information. The information regarding sequences of proteins is transferred through the transcript mRNA and then translated into the sequence of proteins at ribosomes in cytoplasm. 3 nucleotides are used to code one amino acid, and there are specific codes for all the 20 amino acids. Other significance of amino acid sequences. Oligopeptides or short sequences are used to find out unknown sequences of protein or protein domains by using protein database on the basis of homology. In the figure below, short sequences of about 15 and 9 amino acids are shown in green and is used to search the SH2 domain of protein in the protein database through bioinformatics tools. An SH2 domain of 100 amino acids of human and SRC protein of Drosophila is compared. Exact match between these two are shown with a yellow row. Similar but non-identical are used as plus and mismatches are blank. Exact match is shown in green and both are aligned in red color. With the help of known amino acid sequences, one can elucidate mechanism of protein's function or it may be catalytic action of the enzyme. By manipulating or generating specific kind of sequences, one can find out a novel or more useful protein. Many fatal diseases like sickle cell anemia and cystic fibrosis occur due to alteration of a single amino acid within a sequence of protein, which shows the direct relationship between sequence and three-dimensional structure of protein, which means functionality. Now, sequence determination is a part of molecular pathology, and many fatal diseases could be cured through correction in sequences by genetic engineering tools. Proteins have a common ancestor resembling their amino acid sequences. With this fact, evolutionary history is found out. Naturally occurring peptides and their applications. Cell signaling. They help to translate and deliver signal, meaning biological messages, of about molecular structural, cellular functional or effects, that is, acts as agonists, antagonists, modulators, mediators, hormones, cofactors, effectors, stimulators or activators, etc. Metabolic regulators. Many peptides act as a regulatory agent like enzyme inhibitors, antimicrobials, etc. They can easily travel across the membrane bilayer and therefore may also interfere with the metabolism. Application in human diagnostics and therapy. They may act as a potential source of human diagnostics and therapy because of their small size, low immunogenicity, high stability and functional role. Few examples are given below of the potential of peptides in therapeutics. Therapeutic effects of glucagon like peptide 1, GLP-1 agonist in the treatment of heart failure due to ischemia was studied by K. Wong group, and they found that GLP-1 agonist acts as a modulating agent in cardiac muscle metabolism and improves cardiac energetics. Radio-labeled peptides can be used for monitoring the efficacy and duration of antibiotic treatments. Poisonous and venomous animals secrete venom fluids which contains mixture of complex compounds like modified peptides by post-translational amidation or phosphorylation. These peptides have high affinity and specificity towards targeted enzymes, ion channels, receptors, etc. T. Kubo's group from Japan identified variety of venom bioactive peptides by using clones of cDNA library isolated from the Chilean common tarantula Grammostola rosea venom gland. Through these methods, they were successful in identifying 48 novel toxin-like peptides. After analyzing the cDNA sequences of about 1500 clones out of 4000 clones, and among them 24 toxins are ICK motif peptides, 11 peptides are MIT1 like peptides and 7 are ESTX like peptides. Synthetic peptides have the following applications. Protein sequencing through degradation method, in which verification of small peptides are generated after peptide lysis, is done by synthetic peptides with known sequences. They are also used to deduce the correlation in between biological function of proteins or peptides and their structure and illustrate possible molecular mechanisms behind that. Lastly, these are also used to explore and design new therapeutic drugs like peptide-based immunogens, hormones, vaccines, etc. Students, we have now come to the end of this lecture. Let us do a brief summary of what we have learnt today. The primary structure of proteins is a straight chain of polypeptide chain made up of amino acids and linked with peptide bonds, which are covalent in nature. We have also learnt about the characteristics of peptide bond and the fact that the structure of the protein depends on the sequence of amino acids in the polypeptide chain. The polypeptide chain length may vary from 2 to 6 lakh amino acids, and up to 50 amino acids it is known as peptide, and above this it is called a protein. The folding of protein in its native form depends on the properties of the side groups of amino acids present in the polypeptide chain. naturally and synthetic peptides play important biological roles and can be used for therapeutic and drug development purposes. We hope you found this lecture useful. Thank you for watching this lecture on primary structure of proteins.